I have the honor of kicking off and opening the meeting with by introducing our first keynote for today, who's made the trip all the way from California to be here. So it's my pleasure to introduce Dr. Marianne Bronner. Dr. Bronner is the Edward B. Lewis Professor of Biology at Caltech, and her lab has made seminal contributions to the field of developmental biology, for which she has received numerous awards and honors, including the Edwin Conklin Medal from the Society of Developmental Biology. She was elected to the U.S. National Academy of Sciences in 2015. And her work has advanced our understanding of the molecular mechanisms that underlie the formation and the evolution of the neural crest, which is, I think, as many people here know, a highly multipotent stem cell population that gives rise to melanocytes in the skin and the craniofacial skeleton and peripheral ganglia. Dr. Bronner's lab has been central to efforts to define a deeply conserved gene regulatory network that controls neural crest development. And her lab uses a comparative and multi-organismal approach to studying features of the neural crest and its development that are shared across vertebrates from, lampreys to frogs to chickens to us. So please join me in welcoming Dr. Bronner to the stage.
Thank you very much, Cole, for a great introduction, and I really appreciate being invited by all the organizers to this great meeting. I am from Southern California, so it's a bit of a shock to be in this weather, but I'm hoping it's going to improve. So first of all, I wanted to tell you a little bit about the neural crest and why I think it's such a fascinating cell population. Cole has already told me he's really disappointed I'm not going to talk about lamprey, but I will talk about other vertebrates. And so this, I just wanted to introduce the neural crest by how beautiful it is. This is an image of a chick embryo, and neural crest cells arise as the nervous system is forming in the midline of the embryo, and then they migrate away, and they undergo some of the most extensive migrations of any embryonic cell type. So here's an embryo in which we've electroporated a GFP construct into the neural tube, and you can watch these cells as they move from the central nervous system into the periphery. Due to the pioneering work of Nicole Ledouin and colleagues, we know a lot about the derivatives that the neural crest forms by her lineage analyses. And the thing that's quite remarkable about this stem cell population is the diversity of derivatives to which it gives rise. So not only, as the name implies, does it give rise to much of the peripheral nervous system, but in addition, neural crest cells give rise to the pigmentation of the skin, much of the craniofacial skeleton, and different types of ganglia. So there's really great diversity in the derivatives that come from neural crest. So what I'm going to focus on today is the fact that not all neural crest populations are the same. So if you look along the body axis, there are different subdivisions of the neural crest, and they differ in terms of the derivatives to which they give rise. So just as an example, only the cranial neural crest that arises in the head of the embryo has the ability to form bone and cartilage, whereas other populations of neural crest cells appear not to be able to do so, even if they're transplanted to the right region of the embryo. So what I'm going to focus on today is the diversity of these cells along the body axis, particularly focusing on the neck region of the embryo. And so this just shows a map of the types of derivatives that come from neural crest. I've shown you that the cranial neural crest forms craniofacial cartilage, but these vagal neural crest cells in the neck region actually form a great diversity of derivatives. They give rise to parts of the heart, including the outflow tract, and they also give rise to enteric nervous system, which is the nervous system that innervates the gut. And so that's what I'll be focusing most on today. I have to admit that I do use single-cell biology, but you may not hear the word single cell until about a third of the way through my talk. So sorry about that for those of you who might be disappointed. So this is a general outline of what I'm going to talk about today. First, I'm going to focus on the cardiac neural crest. These are the cells that contribute to parts of the heart. And I'm going to tell you a little bit about the gene regulatory mechanisms that we think make these cells unique. Then I'm going to talk a little bit about left-right asymmetry in this cell population. And it's well known that the heart is left-right asymmetric, but we only recently discovered that there may also be left-right asymmetry in the neural crest. And finally, I'm going to focus on the cells that give rise to the enteric nervous system. So first, let's start with the heart and the cardiac neural crest. Okay, so the neural crest that arises in this neck region of the embryo is quite amazing because these cells migrate from the hindbrain region, and then they migrate from the neural tube, and they wait until the heart begins to form. So the heart forms, these cells pause, and then once the heart forms and begins to connect to other tissues, these cells migrate into the outflow tract of the heart. And they are very important, and some beautiful work done by Margaret Kirby in the 1980s and 90s really explored these cells and how important they were for heart development. And one of the first experiments she did was to surgically remove this cardiac neural crest population. So we repeated this experiment as shown here. So this is a cross-section through the neural tube, and these are the cardiac neural crest cells. What Kirby found is that if you removed one of the neural folds that contain these neural crest cells, or both of the neural folds, you ended up getting a very severe heart defect. So the heart connects to the lungs by a blood vessel called the aorta, and what the neural crest cells do is they come in and they form the septum that divides the aorta into an aortic and pulmonary trunk. So that's where the blood goes from the heart to the lungs and then gets oxygen and then comes back. So if you remove the neural fold, you get a hole in the heart, so the septum doesn't form properly, and you get an even more severe phenotype if you ablate both sides of the neural crest. So the other thing that Kirby found was that other neural crest populations, either the cranial neural crest or the trunk neural crest, if transplanted in place of the cardiac neural crest, were unable to rescue this phenotype. So in other words, if you put the trunk neural crest in place of the cardiac neural crest, you still got this outflow tract defect. And interestingly, if you grafted the cranial neural crest in place of the cardiac neural crest, those cells not only gave you this phenotype, but they ectopically formed cartilage, which is their normal derivative. So despite the fact that these cells look so similar to one another in terms of their early properties, they clearly have differential abilities to give rise to cell types. And we were fascinated by how that might happen. So my student, Shah Gandhi, decided to tackle this question, and basically we did this initially by bulk RNA-seq, and so basically you can take out a population of cardiac neural crest cells that we label with a fluorescent enhancer and isolate those, and we have another enhancer that labels the trunk neural crest, and then we can compare them and look at gene expression differences between the cardiac and the trunk neural crest. And as you can see, we saw a lot of genes that were enhanced in the cardiac crest population, and this is Shah who did this work. So the first thing we do is just look to see where these genes are. And one of the surprises we got is that many of the genes that we isolated were not only expressed in the cardiac neural crest, but were shared between the cranial and the cardiac neural crest, though we did find some cardiac crest-specific genes. So this is an in situ hybridization, where this is the neural tube and the hindbrain is here, this is the ear. And the cardiac neural crest cells migrate from the hindbrain and then migrate along this path to the heart. So this gene, MAFB, was expressed uniquely in this cardiac crest population. But other genes like this were expressed in both the cardiac and the cranial neural crest. So we were particularly interested in one gene. This gene had a beautiful expression pattern in this cardiac neural crest. And we discovered it on Friday, so we called it TGIF. Not really. But what was exciting about it is that when we looked at the earliest expression we could find of this gene in this hindbrain region that would form the cardiac neural crest, we found that it initiated expression at exactly the time that we knew the cardiac neural crest became different from either the cranial or the trunk neural crest. So we were particularly interested by looking just at an expression pattern at what this gene might be doing. So the first thing we do is we knock out the gene, and we use CRISPR-Cas9 technology. So this is an early chick embryo, and so the experiments I'm going to show you this first part of the talk were all done in bird embryos. And the reason we do this is because the heart in the avian embryos is extremely similar to the human heart. So this would be a gastrulous stage embryo. This is the midline. And the nice thing about bird embryos is they lie as a disc on top of a large yolk. So they're very easy to manipulate. And we can put a CRISPR-Cas9 construct into one side of the embryo and then use the opposite side as an internal control. And this is really nice because it allows us to compare control and experimental in the same embryo. So this is what it looks like. So we put the TGIF CRISPR on the right, and we put a control on the left. And when we knock out TGIF, we lose TGIF as we expect. But what was most interesting is that all those other cardiac crest genes that we found were also downregulated when we knocked out TGIF. So that was exciting, and we thought maybe this was a really important gene for giving the cardiac neural crest its unique identity. So the next thing that we do is we wanted to do a knockout, a global knockout of this gene. And the way we do this is basically rather than putting CRISPR on just one side, we put CRISPR on both sides of the embryo. And then in order to do this in a way where we can recognize the cells in which the knockout was done, we actually often do this in a GFP chicken, and then we transplant the neural folds in place of a wild-type neural fold. And what I like about this experiment is it really takes advantage of modern technology, CRISPR technology, but also old-fashioned experimental embryology. And to those young people in the audience, I want to stress that we have all these wonderful techniques now, but it's also good to keep some of the old techniques. And I think there's power in combining the two. So we knocked out, this is a control, so we knocked out TGIF and compared it to the control. So this is the outflow tract of the embryo with the aorta and the pulmonary arches. And as expected, we saw our green neural crest cells migrating to form the septum of the heart. We were very excited to see that when we knocked out TGIF, these green cells actually went to the right place. So they migrated normally, but they failed to give rise to this outflow tract septum. So that again suggested that TGIF was a really important gene. And so I hypothesized at this point that maybe that's all you need to make the cardiac neural crest different than other neural crest populations. And as most of my graduate students will tell you, when I formulate a hypothesis, it's usually wrong. And in this case, TGIF was not sufficient to cause other neural crest populations to be like the cardiac neural crest. So we reasoned that perhaps there were other genes that cooperated with TGIF. So okay, get ready. I'm going to say single cell. So we went to our single cell data. We did SMART-seq of the cardiac neural crest, and we looked for genes that were co-expressed together with TGIF. And what we found is that a transcription factor SOX8 that we knew was very important in neural crest development was expressed together with TGIF, as was ETS1. And there were other genes that were co-expressed as well, and I'll show you those in a bit. But we reasoned that perhaps these three genes might be sufficient because we knew from Yamanaka's work that 3 genes were sufficient to reprogram skin cells to become like embryonic stem cells. So we thought maybe 3 would be enough. And so we basically went back and did an experiment to test to see what would happen if we took these 3 genes and put them into another neural crest population. Could we give them a cardiac crest identity? So basically here we go back to our transplantation experiments where we can look to see what happens with a cardiac neural crest transplanted in place of the endogenous cardiac neural crest. Or we can take a trunk neural crest and put it in place of the cardiac neural crest, which we know does not normally rescue heart defects, but if we reprogram it with these 3 genes, we wondered if that was sufficient to give it a new identity. And so what we found is this is the control, again, where we have these cells going to the outflow tract septum. We repeated this experiment that Kirby had done years ago, where she took the trunk neural crest and put it in place of the cardiac neural crest and ended up getting this heart defect where there's the septum did not close properly. But if we simply reprogram these trunk neural crest cells with these 3 transcription factors, that was sufficient to rescue this phenotype. So that was very exciting for us because it suggested that these 3 genes were enough to give a trunk neural crest cell a cardiac neural crest cell-like identity. So basically what I've shown you is that we have this, what we call a cardiac neural crest subcircuit. And if we remove just one of these genes, TGIF, that's sufficient to sort of knock out this ability for these cells to form a normal cardiac neural crest derivative. So then our next step was to see if, in fact, TGIF is important, how does it work, and what might be downstream. So here we turn back to our SmartSeq data, and we found that TGIF also was expressed with a number of other transcription factors, TWIST1, FOXC2, and FOXP1. So here we took a bit of a dive, and we wanted to know if, in fact, there's co-expression, might these things be actually turned on by TGIF? And so we basically just tested all these three factors to see what would happen if we knocked them out individually. So we went back to our embryology, and we did our CRISPR knockouts on both sides of the embryo, transplanted the dorsal neural folds in place of a wild-type neural fold, and just saw what would happen. So here again is our control, where we have an aortic and a pulmonary septum. And our labeled neural crest cells have gone all over the heart, and as they normally should. However, when we knocked out one of these genes, TWIST1, what we found is now the septum did not form properly, even though the neural crest cells went to the right place. And when we looked to see what they were doing, when we labeled them with a marker for neurons, we found that these cells that are supposed to be mesenchymal cells of the heart now had turned into neurons. So we actually changed their fate by knocking out TWIST1. We knocked out these other two genes as well, FOXC2 and FOXP1, and all of them gave us this septal defect. So that suggested that these genes may well be working in a circuit together with TGIF and are all required for normal formation of the heart. So this was exciting to us, and what it showed us is that these genes were really important in heart development. So this is all done in chicken embryos, so we wanted to see if this actually had any medical relevance. So we went to the Human Genome Database, and there you have a lot of genes that are involved in congenital heart defects. And we cross-referenced these with genes that might be involved in septal defects in humans, as well as things that were important in development of the cardiac ectomesenchyme. And when we put these three things together, we found there were 11 genes in common. And we were very excited to see that these three genes, FOXC2, FOXP1, and TWIST1, were 3 of the 11 genes that had been implicated in these kind of defects in humans. So I really like this because it suggests that looking at a model organism like a chick embryo might actually give us insights into what may be important not only in bird development but also in human development. And we're really curious to see what the other 8 genes that intersect with this might also be. Okay, so what I've shown you so far is that we have this cardiac neural crest subcircuit that consists of these 3 genes, SOX8, TGIF, and ETS1, and that seems to confer upon other neural crest populations like the trunk neural crest the ability to form heart muscle, which is something they normally cannot do. Well, a few years back, we actually were really interested in the cranial neural crest, and we did a very similar type of experiment that I showed you to look to see what was important in cranial neural crest development. And what we found is these 3 genes conferred upon the trunk neural crest the ability to form cartilage, which they normally cannot do. And if you look at these 2 subcircuits, it doesn't take very long to see that they're very, very similar and in fact share these 2 genes. And the only difference is this AP2 beta is important for the cranial neural crest, whereas in the cardiac neural crest we have now TGIF. So we speculated that maybe there might be small differences that were different between these populations that are actually very close together and share a lot of gene expression. And so we wanted to explore that. So what this experiment where this cranial crest had been done was basically done by sorting either the cranial or the trunk neural crest. And when those 3 genes were then put into the trunk neural crest, these cells were transplanted to the head, and they were normally they're unable to give rise to cartilage, which is shown here in red. But if you reprogram them with these 3 genes, then you get cartilage formation. So this experiment is very similar to what I showed you with the cardiac neural crest, really just differs by this single gene. So we speculated that perhaps that perhaps small differences in transcriptional regulation might be important in conferring large differences in what the cells could give rise to. So we wanted to test that, and the way we did that is the way many of you are doing this, by doing ATAC-seq of the cardiac neural crest, and we're doing this at both the bulk and single cell level, and looking for differential enhancer expression between particularly cardiac and cranial neural crest cells. So what we did is compared many different stages of development, looking at pre-migratory cardiac neural crest and migrating cardiac neural crest, and just looking for enhancers that were open in this population. And we were particularly interested in finding enhancers that might be important and different in the cardiac neural crest compared to the cranial neural crest. So we found many enhancers, as one does, and we found some that were uniquely expressed in this subpopulation. So these are, again, are cells that are expressed in the hindbrain and then migrating from the hindbrain into the heart. But we were a little bit disappointed because many of our enhancers, in fact, the majority, were not unique to the cardiac neural crest, but were rather shared between the cranial and the cardiac neural crest. So we started delving through these different enhancers, and I'm just going to show you one example when we compare the cardiac neural crest and the cranial neural crest of an enhancer that was open in both populations, as shown here, versus one that was uniquely open in the cardiac neural crest. And both of these enhancers were just upstream of CXCR4, which is a chemokine receptor that's known to be very important for neural crest migration. So when we looked at these enhancers in more detail and we looked for transcription factor binding sites, what we found is that in this shared enhancer, we had an AP2 beta binding site, whereas in this enhancer, which is unique to the cardiac neural crest, we had a TGIF binding site. So we were curious as to whether these binding sites might be important and whether perhaps a duplication in this enhancers and then divergence might have given us a new identity that promoted cardiac neural crest development. So this is the expression pattern of this enhancer that is shared between the cranial and the cardiac neural crest. Versus this enhancer, which is unique to the cardiac neural crest. And so I won't show you the data for this, but what we found is if we delete this transcription factor AP2 beta binding site, what happens is we lose this cranial expression, but we retain the cardiac expression, suggesting that this binding site is important for expression within the cranial neural crest. And so what we wonder is if this is something, these enhancer binding sites might have emerged and made one population into two separate populations. So basically, losing this binding site causes loss of this cranial enhancer. And in other enhancers, we found that loss of TGIF1 causes loss of expression in the cardiac region. So what these data I've shown you suggest is that this cranial versus this cardiac neural crest population may have diverged from one another by perhaps new and small regulatory changes that change the fate of the cells from giving rise to bone and cartilage to basically another type of ectomesenchymal cell type, which is this outflow tract smooth muscle. So it's basically a change from bone and cartilage to smooth muscle by small regulatory changes. And that's a speculation, but one that we're really interested in pursuing. Okay, so just to summarize what I've shown you, we think that there's this important cardiac neural crest subcircuit that has emerged, and that has helped confer unique identity onto the cardiac neural crest. So one of the things we always assumed with all neural crest populations is that they're pretty symmetric. And in fact, I showed you that we take advantage of this property by doing knockouts on one side of the embryo versus the other side. So part of the work that I showed you just now has been published. And one of the things that we had a really nasty reviewer ask us is, why are you always doing your knockouts on the right side of the embryo and using the left side as a control? And I thought this was pretty stupid. And we had done the same thing with our ablations. We were always ablating on on the right side and using the left side as a control. So just to satisfy this reviewer, we decided we had to repeat some of our experiments. And so we basically took this ablation experiment I showed you, where we did our right ablations, and we decided we had to check and see what would happen if instead of using right-only ablations, we did left ablations and compared the two. And as you know, we always complain about reviewers, but sometimes they actually come up with interesting ideas. So we decided to really test this in some detail and look to see what would happen if we did transplantations between right and left and right, and ablations between left and right. So what we found when we did our transplantations, and this is transplanting left to left or right to right, you actually see different contributions from the left versus the right cardiac neural crest. These are the aortic arch arteries, and what you can see is when you transplant a GFP-labeled left neural fold, you really label the left aortic arch arteries, whereas if you do a right transplantation, you label the right aortic arch arteries. So maybe it wasn't so stupid to think about what the differences might be. So we basically found that these cells seem to stick to one side, but once they got to the septum, between the aortic and pulmonary trunk, that was pretty uniform. So we went back and we decided to test what would happen if you ablated the left neural crest or if you ablated the right neural crest. And then these experiments are really hard, actually, because it's really hard to keep embryos alive after doing surgeries like this. But I have a wonderful postdoc in my lab, Tatjana Solovieva, who does wonderful experimental embryology. And she basically did a lot of experiments, got these embryos to survive, and looked to see what the phenotypes were. And normally you get this nice septation between the left and right side, but if you ablate the neural crest, you get this defect that I've shown you, where you have basically this hole between the heart. But when she did the quantification, what she found is that in the left case, it looked pretty normal, and she got many more defects when she did right neural crest ablations. This was particularly true in the aortic arch arteries, and I know this anatomy is really hard to sort of get your head around. So when we were walking in the park one day, we saw this nice tree, and this is a better way to represent the aortic and the pulmonary trunks. This is what they look like when they're connecting to the lungs. And so if you look at the anatomy and the types of defects that you get, what you find is that in the aortic arch arteries, they're pretty normal after left ablation, but the right ablation causes either fairly severe abnormalities of the arches, and it also causes different orders of fusion, mostly from the right ablations, not from the left. And so perhaps one of the reasons we got such good phenotypes is we mostly did right ablations. So what's very interesting about this is that it suggests that there are real differences between the left and right side. And so the next thing we did was ask what would happen if you switched left and right, basically grafted the left to the right or the right to the left. And what happens when you do that is you really mess up heart development. So basically, these neural crest cells went all over the place. There were many fewer of them from right to left or left to right ablations, and these hearts had many more abnormalities. So that suggested that perhaps there were, even in these very early neural crest populations, intrinsic differences between them. So now what we're doing, I should mention these are sort of hot off the press experiments, but what we've been doing is beginning to do bulk and single cell RNA-seq, and what we've discovered is that there's a very narrow time window at which the cardiac neural crest really emerges as being different. If you look at what we call an early time, which is just as the neural folds are beginning to close, the two populations are very similar to one another. But at the 10 somite stage, which is just when the neural crest is beginning to migrate, you really get distinctions between the left and the right cardiac neural crest. And from looking at a volcano map, we begin to see genes that are highly upregulated in one side or the other. And we've been starting to do a hybridization chain reaction to look at the expression of these genes. And this is one example of a gene, PAL5F3, and it's highly upregulated, well, no, it's upregulated slightly in the left versus the right cardiac neural crest. So we're just exploring these changes now, but what we speculate is perhaps that slight asymmetries in gene expression may actually be important in conferring identity to these cells, and that there are intrinsic differences between these two populations that we thought were very similar. So just to summarize this part of the talk, which is really very preliminary still, we're very excited that we seem to have a much more defined defect from a right cardiac neural crest ablation than a left, and we speculate that prior to neural crest migration, there may be intrinsic differences, so these cells already know where they should go and what kinds of things they should do. Okay. All right, so I'm going to end my talk by just telling you about a little further down the body axis, which is a population called the vagal neural crest, and this gives rise to the enteric nervous system. However, if you look along the body axis, this is in this diagram that I've shown you, I've talked so far about the cranial and the cardiac neural crest. The vagal neural crest is just below that, and the enteric nervous system forms largely from this vagal neural crest population. However, in amniotes, animals that have come onto land, there's a second population called the sacral neural crest, which is at the very bottom of the embryo, and these cells also contribute to the enteric nervous system. And the way the enteric nervous system forms is that these cells from the neural crest migrate into the body, and then as the gut develops, as the intestines develop, they migrate all the way down the body to form different types of neurons of the enteric nervous system. This additional sacral population migrates oppositely. It migrates into the gut and then up to just the post-umbilical region, and then it stops. So there's mixing of the vagal and the sacral neural crest cells in this part of the embryo, but this top part is derived only from the vagal neural crest. So the enteric nervous system is an amazing part of the nervous system. It's often called a second brain because it's so complex. It has about 500 million neurons in humans. And so these neurons are in the form of two plexuses, the myenteric plexus and the submucosal plexus, and they control peristalsis in the gut. Those of you who had a big lunch, your enteric nervous system is working hard. You're probably going to sleep right now. And so it's very important. So we really wanted to, one of the challenges in looking at the enteric nervous system is trying to understand the contributions of these different neural crest populations. And indeed, almost nothing has been known about the various contributions of the vagal versus the sacral neural crest population. So we wanted to go back and do some lineage experiments to try to actually understand the relationship between these populations. And this is another example of why bird embryos have some advantages. There's some viruses that are avian specific, and many years ago, Connie Sepko's lab actually modified an RCAS virus and made it into a replication incompetent virus by removing the envelope protein. So one of the things we've done is taken this virus and actually manipulated it more and put in multiple cloning sites that enabled us to basically create a whole library of viruses that have different expression patterns. Some are expressed in the nucleus, others are expressed in mitochondria, others can be expressed in the membrane, and basically we can use these to label different neural tube and neural crest populations. So this is what these different viruses look like if we inject them into the neural tube, and we can actually do lots of clonal analysis with this, which is not what I'm going to talk about today. I just like to show this because it's really pretty, but this labels not only the neural tube but also the neural crest cells that migrate out from the neural tube. So I had two wonderful students working on this project, Lily Tang and Jessica Jacobs-Lee, and what they did is they selectively labeled in the same embryo the vagal neural crest or the sacral neural crest population. So you can label one in red and then let the embryo develop until the sacral region forms and label the other one in green. And then we could actually compare what these two populations give rise to. And what we found is that the two populations actually generally segregate from one another and give rise to some overlapping, but also some different cell types. So for example, this is the ganglion of Vermaak, and this contains many sacral neural crest cells, but very few of the vagal population. And some of the neurons that differentiate seem to be unique to the vagal neural crest. So of course we wanted to look at this by single-cell RNA-seq, and so we can isolate separately the vagal population either from the the rostral part of the gut, where there are no sacral neural crest cells, or the caudal part of the gut, where there are both vagal and sacral neural crest cells. And what we found was actually quite surprising. We expected the vagal cells to all be relatively similar, but indeed what we found is the vagal postumbilical and the sacral postumbilical were much more related to one another than the preumbilical vagal cells. And so one of the striking differences we found is that this population was a precursor pool that seemed to be missing from this sacral population. In contrast, this population, which is a mostly glial cell population, was pretty much depleted in the vagal population, in the preumbilical vagal, but shared between the postumbilical vagal and the sacral. And finally, different classes of motor neurons seem to be unique to this vagal preumbilical neural crest, expressed a little bit in the postumbilical, but absent from the sacral. This work was done at so far a single time point, so now we're doing this at multiple time points. But this allowed us to, in the same group of embryos, selectively label one of these different populations. So basically, this was very useful for looking at the vagal versus sacral neural crest, and it pointed out differences not only in the ability to give rise to different types of neurons, but also differences in the precursor population between these two populations. So this work is ongoing, and now we're doing multiple time points, and so the goal will be to look to see how differences arise in these populations over time. So there is one limitation, however, in using any of these amniote species. So this was done in birds, where we were able to do this nice lineage labeling, and there have been many experiments done on the enteric nervous system in mammals, but they're usually limited to a very particular part of the gut because it's so hard to get the, different cell types from different regions. So one of the things that I'll end my talk on is that we really would like to know what happens along the entire length of the gut. So to do this, we actually turned to a different system, and that is the zebrafish. So zebrafish has many advantages in that they're transparent, so they're very beautiful and clear, but they have a simplified enteric nervous system. So whereas in amniotes, the nervous system is more complex, many more neurons and big ganglia, there's also a myenteric and a submucosal set of plexuses. In contrast, in zebrafish, there's only myenteric neurons, and they're scattered along the entire length of the gut. So we decided to look at this, and a wonderful graduate student in my lab, Sun Lee, has been doing this work, and she reasoned that we needed to have a very good transgenic line in order to isolate these cells. So she took this transcription factor FOX2B and wanted to label those cells particularly because they would be a very good population for isolating pure populations of both neural crest precursors as well as differentiated neurons, since FOX2B is expressed in all of those populations. So what Sun did was she directly tagged this FOX2B gene with a neon green marker and created a transgenic cell line in zebrafish. And so this just shows this marker, which labels beautifully all these FOX2B positive cells, and she could then follow them over time. And if this movie plays, what you can see, this is a ventral view of these cells migrating along the gut of the embryo. So they start out in the foregut, and then they migrate as two streams down both sides of the gut. So using this marker, Sun was able to isolate pure populations of both precursors and neurons from the guts of zebrafish at multiple time points. And in fact, she did the most exhaustive set of single-cell experiments, looking at 2, 3, 4, 5, and 6 days of development. And by 6 days, these zebrafish larvae are already feeding, so the enteric nervous system is fairly mature. Okay, so this is what the single-cell map looks like, and you can isolate it into all these different days to get an idea of how the cells are differentiating. And when we looked at gene expression, we were able to take these different clusters and get an idea of what kind of identity the cells had. And so in so doing, this population here is a precursor population, but she found 3 distinct populations of neurons, serotonergic neurons here, inhibitory motor neurons here, and excitatory motor neurons here. And so then what we can do is, of course, RNA velocity analysis as well as tree analysis using SC fates to try to get an idea of all the different types of neuronal populations that develop and how they segregate over the course of time. And so this allowed us to build a lineage tree. This just, I like this, you know, you can make these cool rotating movies and you can get an idea of all these different endpoints. But what was most informative for us is that we could start out with the early neural crest population. We got different precursor populations that formed. This then branched into a tree that would give rise to different types of neurons. Some of these were excitatory neurons, some of them are inhibitory neurons, and we could actually look at their lineage relationships. So this is another way to represent that I think is a little bit easier to see. And what was really interesting to see from the lineage analysis is that these serotonergic neurons seem to branch off first from all these other types of neurons. Then we got excitatory motor neurons branching off, and then various subtypes branching off, and then inhibitory neurons coming here. So what we were particularly interested in is wanting to know what types of factors might be important in really controlling these lineage decisions. And so we took it and we could look at it to see where they were expressed in the gut. So one of the beautiful things about zebrafish is that you can actually analyze the whole gut and look at their expression patterns. And we can do sort of a modified form of spatial transcriptomics by using hybridization chain reaction, and we can identify up to 13 transcripts or more in this way. So this is the entire length of the gut in zebrafish. We labeled them with different neurotransmitter markers, and then we can look at different subcategories. And you won't be able to see anything here, but basically what we were able to see is where the different subtypes were all along the body axis. And I think it's the easiest way to see this is here. These colors just represent different subtypes, and we have some that are expressed all along the body axis. Others are expressed just in the caudal part. Others are expressed in the rostral part. But I just wanted to focus on this particular peptide, CART2, that was expressed only in the very posterior part of the gut. So that was very segregated from other parts of the gut. So in looking for genes that might be important in establishment of that CART2 population, what we found is that GATA3 aligned nicely with where those cells were located. And when we looked at the expression pattern, CART2 is only expressed in this very caudalmost part of the gut, whereas GATA2 is expressed everywhere except in these CART2 positive cells. So we wondered if it might be negatively controlling CART2 expression. So basically what we can do is a CRISPR knockout, similar to what I showed you in chick, except in zebrafish, we do this at the one cell stage, and we use 3 different guide RNAs to knock out each gene. And what we found, as a control, we used tyrosinase. So this is the normal expression of this. This is a transporter important in excitatory motor neurons, and you can see it's expressed all along the body, whereas CART2 is only expressed here. When we knock out GATA3, what we found is we completely lost this type of neuron, whereas CART2 was now distributed throughout the gut. So that suggests that GATA3 is expressing this particular lineage and causing a lineage transformation where this type of neuron is lost and CART2 is enhanced throughout the gut. And so we can do similar types of experiments with other genes that we think are important in different lineage decisions. This is a transcription factor EBF1A that's co-expressed with many neuropeptide genes, including VIPB and NOS1. And when we knock this one out, what we find is that this is the control where we see NOS1 expression and VIPB. When we knock out EBF1A, we lose NOS1 and we lose VIPB, but we keep expression of other things like VIP. So, oops, that's me. And finally, I'm almost done. SATB is another transcription factor expressed in a different lineage. And again, when we do the knockout, what we find is that this is important in serotonergic neurons. And here's a serotonergic marker that's completely lost when we knock out this gene. So the reason I wanted to show this is that by doing our single cell analysis and then lineage trajectory analysis, what we find is that these 3 genes seem to be important in 3 separate lineages. This one in inhibitory motor neurons, this one in excitatory motor neurons, and this one in serotonergic neurons. So I really like this kind of analysis because it shows how you can dive deep into your single-cell data and look for candidates that might be important in really controlling lineage decisions. And we can even find some that affect specific subpopulations of cells in only particular parts of the nervous system. All right, so just to conclude, I've shown you a lot of data, but basically I'm very excited about how neural crest populations are different along the body axis. And I started my talk by showing you about cardiac neural crest cells, where we found that this transcription factor, TGIF1, seems to be a very important control gene for conferring cardiac identity. I've shown you that we think changes might have occurred over the course of evolution, where small changes in transcription factor binding sites and enhancers may have helped to give this axial level specific identity. I've shown you that there are some left-right differences perhaps in some of these populations that we had previously not appreciated. And finally, I ended up by showing you something about delving deep into neuronal cell lineage decisions in the vagal and sacral neural crest. So I just wanted to acknowledge the people who've done this work. The cardiac neural crest work I showed you in the beginning was done by Shah Gandhi with help from Appy Raja and Max Ezen. The left-right asymmetry is being done by Tatiana Solovieva, and the vagal neural crest work is done by Sun Lee, Jessica Jacobs-Lee, and Lily Tang. And finally, I just wanted to end by inviting you all to Puerto Rico, where the International Society for Developmental Biology is having a meeting in June of next year. And I'm president of that society, so that's why. Okay, I'll end by this. Thank you.
Okay. Uh-oh.
I'm sorry I
Do we
didn't
have
talk about
questions?
Lance.
Oh,
Okay.
come on.
Okay, very nice talk. Thank you. Just a quick question. So, do you
Oh, be sure to use the mic, please, so that the Zoomers
Sorry.
can hear.
Very nice talk. So I have a question about this pre and post umbilical difference in the vagal neural crest. A, do you think it's something cell intrinsic and they are different populations pre-designated for those regions? Or do you think it could also be something to do with the timing? So because you're taking a single snapshot, do you think that they could be in a kind of a later developmental state, pre-umbilical maybe, or something similar? And if you take a different snapshot, you'll see that they've actually caught up.
Yeah, I actually think it's, I don't know if it's largely due to environmental factors, is my view. I think these cells are fairly undetermined when they're beginning their migration, and then depending upon where they land, they listen to the signals that are involved. And that was a surprise. I actually expected to see sacral neural crest cells being quite different than vagal, and in the end, they look quite similar in terms of what they can give rise to. So one of the things we're interested in exploring is what environmental factors and maybe what receptors might be present on those cells that can respond to different signals. So that's one of the things we're doing. But the most important thing now is to do different time points and see if those changes, do they become more pre-umbilical like over time, just like you said. So we don't know the answer to that yet.
Thank you very much.
I can't hear you. Oh, there we go. The lights are.
I know, the
Make
lights
it
are
hard to
on.
see.
Thank you very much. It's a super interesting talk. About the part 2, I'm wondering, because it looks as if when you try to swap the left and right cardiac crest neural crest cells, as you've shown, is a bit messed up in terms of the structure. Do you know whether it's because it looks as if that the cell was already there, decides which cell they're interacting with, either left or right is being defined by the cells that are already sitting there. So I was wondering if there are any or have you been trying to look at the cell-cell interaction between the cells sitting there and also the swapped cells you've done?
Yeah, well, we haven't, these are actually quite new experiments, so we haven't done too much with it. But one of the things I find most intriguing is the fact that there's a very sharp time point at which these differences arise, and it's exactly the same time point that we know that the cardiac crest becomes different from cranial or the trunk. And so we think there's some inherent transcriptional changes that might be occurring, not so much to do with environmental factors, but likely in the cell, somehow in the cells themselves. And I guess one way to answer your question would be if we did the swap before that time point, would they now have the correct identity? And if that were the case, it might be that there's some factors coming from there is left-right asymmetry in the embryo. Maybe they're being signaled at that particular time point. But what we think is that at least by the time they're fixed, which is at this 10 somite stage, then something really is different. So that's what we're exploring. But to me, it was just a big surprise because we hadn't anticipated this at all. Yeah, so ongoing. I'll let you know.
Fantastic talk. Really, really enjoyed it. Don't know much about the neural crest, but what really defines these boundaries? I mean, at what point does the cell stops being TGIF and becomes like TFAP2B? Is it? I mean, what is there?
Yeah, that's an excellent question, and one of the things I'd really like to know most. But the boundaries are fairly sharp, and if you look at this TGIF1 gene expression, it's actually quite restricted to this particular region. And so one of the things we don't know. The answer to your question is I don't know yet. But one of the things we're doing right now is trying to look at the difference between cardiac and vagal, which is also very close. So we're doing single cell analysis of those two populations to see if we can see what sets up the border. We do know that the cells that migrate to the heart use a signal CXCR4, and the ones that migrate to the vagal do not use that. They use RET. So there are some differences like that, but those genes are expressed uniformly in both populations, so that's not the answer.
Super quick follow-up. Do you think that pattern is established already in the neural tube before the crest begins to migrate?
I do think that, but I don't have any evidence for it.
Maybe do we have any from the iPod? Nobody has an iPod anymore.
Yeah, I'm afraid the internet is not working.
The internet is not working. Okay, so the answer is no.
Beautiful talk. I have a question about your comment that it's interesting that the same genes are getting repurposed throughout the lineage. Do you think that that reveals the evolution of how these cell types emerge?
We actually know a bit about that. So if you look in lamprey, what you find is that there's a basal neural crest gene regulatory network. And then over time, it becomes elaborated. And so what seems to have happened is that you start adding more complexity to this basal network. But interestingly, at least for some of the cranial genes that we found, they're sort of added uniformly along the body axis, and then only when animals came to land, they got lost from other populations. So I think so we don't know exactly how, and the cardiac crest, or this vagal crest, actually didn't exist at all in lamprey. So there are these evolutionary changes that have occurred, but I think it was simple in the beginning and then became more complex and then diverged even more. Let's see.
Am I missing anybody? Okay, maybe I'll ask one.
Okay.
Moderator's prerogative. So that was well, I have one.
Oh, right.
And I'm wary of some, deafening feedback loop. That was a beautiful talk about the neural crest, so I thought I would ask you about everything aside from the neural crest. Like these experiments where you can either transplant or with a knockout have tissue that is either not invaded by the neural crest or invaded by the wrong kind. Have you done any single cell analysis on the other cells to see whether or not their transcriptomes are messed up for having not received the messages that they're supposed to be receiving from the neural crest?
That's a great idea. No, we haven't. The problem is that there's a lot of tissue there, so it would be difficult to do. But I mean, one of the things that's interesting in terms of neural crest development is this outflow tract probably arises not just from the neural crest, but by an interaction between neural crest and second heart field. So I think it would be really interesting to see how the neural crest cells or their absence changes the second heart field. And that's something that I don't think we could do in a chick embryo, but it might be possible to do in a mouse, where you could isolate those cells with or without the neural crest. We have not done it yet, but I think it's a great idea. Anyone who's interested in heart development, that would be a cool thing to do.
Okay.
So.
Any further questions? All right, well, let's thank our keynote speaker again. That's really still for you, I'm sorry.